Hi everyone, you're watching Educat for NEET. In today's video, we will cover Chapter 11 Biotechnology Principles and Processes. In this chapter, we will learn about principles of biotechnology, on what principles biotechnology is based, and the rules of recombinant DNA technology, the tools. What tools do we need? What are the different equipments we need for recombinant DNA technology? We will also learn the process of recombinant DNA technology, how it is performed. Let's start. Biotechnology deals with the techniques of using live organisms or enzymes from organisms to produce products and processes useful to humans. Means biotechnology is that study or that technology which uses live organisms. For example, in the next chapter you have insulin example. In this example we are using biotechnology. We are using the bacteria E. coli, that is a living organism, and we are producing insulin out of that E. coli. So that insulin is useful to humans. That all techniques are known as biotechnology. These are the techniques of using live organisms or the enzymes from the organisms to produce products and processes which are useful to human beings. In this sense, making curd, bread, or wine, which all are micro mediated processes, could also be thought as a form of biotechnology. Curd, bread, and wine are useful to humans, and these all include microorganisms in this. So, we can say that making of curd, bread, or wine is also a form of biotechnology. However, it is used in restricted sense today is to refer to such of those processes which use genetically modified organisms to achieve the same on a larger scale. But to be specific, we use biotechnology in that form where there is genetically modified organisms. For example, if we look at the E. coli example only, there in that E. coli bacteria we have genetically introduced the insulin gene of humans. So therefore they are producing that insulin which is useful to us with the help of our own gene. We have genetically modified it. Further, many other processes or techniques are also included under biotechnology. For example, in vitro fertilization leading to a test tube baby, synthesizing a gene and using it, developing a gene vaccine, or correcting a defective gene are all parts of biotechnology. Formation of a test tube baby and synthesizing a gene means producing a gene and then using it, and developing a DNA vaccine or correcting a defective gene. These are all the parts and you can say uses of biotechnology. The European Federation of Biotechnology, EFB, has given a definition of biotechnology that encompasses both traditional view and modern molecular biotechnology. The definition by EFB is as follows: the integration of natural sciences and organism cells, parts, therefore, and molecular analogues for produces and services. Means this definition explains as that we are taking the help of natural sciences and organism, naturally existing organisms, as well as the molecular analogues, means the function we are producing through gene manipulation. This is the definition of biotechnology given by EFB for produces and services, means for our own benefits we are producing new products and we are serving ourselves. Coming to the first main topic, principles of biotechnology. Among many, the two core techniques that enable birth of modern biotechnology are genetic engineering. Genetic engineering, let's talk about that first. It means techniques to alter the chemistry of genetic material, DNA or RNA, to introduce these into host organism and thus change the phenotype of the host organisms. So genetic engineering basically means to alter, means change the genetic material, whether it can be DNA or RNA, to alter the existing DNA or RNA and to introduce these into host organisms and thus change the phenotype of the host organism. We are changing the DNA and introducing it to the host organisms, and therefore what is happening as a result? The phenotype, means the outside of the host organism, is changing. The second principle is bioprocess engineering, which means maintenance of sterile microbial contamination free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. We are maintaining a sterile organism, sterile condition in which only one kind of microbe can grow, so that one desired antibiotic, vaccine, or enzyme can only grow. This is known as bioprocess engineering. Let us now understand the conceptual development of the principles of genetic engineering. You probably appreciate the advantages of sexual reproduction over asexual reproduction. The former provides opportunities for variations and formulation of unique combination of genetic setup, some of which may be beneficial to the organism as well as the population. So these were the advantages of sexual reproduction. They help in the formation of unique combination of genetic setup, and most of them are beneficial to the organism as well as the population. While talking about asexual reproduction, these preserve the genetic material. Sexual reproduction permits variation. Traditional hybridization procedures used in plants and animal breeding, they lead to inclusion and multiplication of undesirable genes along with the desirable genes. Sometimes what happens, we breed two plants or animals, they sometimes lead to undesirable genes, accumulation of undesirable genes along with desirable genes. So we cannot specifically take the desired genes. The techniques of genetic engineering, which includes creation of recombinant DNA, use of gene cloning and gene transfer, overcome this limitation and allows us to isolate and introduce only one or a set of desirable genes without introducing undesirable genes into the target organism. So, the most biggest advantage of recombinant DNA is gene cloning or gene transfer, in which we can transfer the desirable set of genes only. For example, in a plant, we want that plant to produce green seeds, but the plant is producing green seeds along with yellow seeds. If we apply this recombinant DNA technology, which involves gene cloning and gene transfer, we can allow the plant to produce only green set of seeds. So this is the meaning of recombinant DNA technology. This overcomes the limitation. Initially what happened, yellow and green, both these seeds were being produced. Now when we introduced this helped us to isolate that gene which produces green colored seeds, and we introduced that genes into the organisms and only green color seeds were being produced. The undesirable genes were not produced, means no yellow seeds were coming in that. So this is the biggest advantage of recombinant DNA. Do you know the likely fate of a piece of DNA which is somehow transferred into an alien organism? Most likely, the piece of DNA would not be able to multiply itself in the progeny cells of the organisms. If we introduce any DNA into an alien, for example an X organism, it will not be able to replicate, because that organism will know that it is not its DNA, its own DNA, it's the foreign DNA. So it will not be able to replicate itself and it will not enter the progeny cells of the organism. But when it gets integrated into the genome of the recipient, it may multiply and be inherited along with the host DNA. If instead of simply putting the DNA inside, we are integrating it with the genome of the recipient, then as the genome replicates, the DNA which we implanted, that will also replicate and it may pass into the progeny. This is because the alien piece of DNA has become part of the chromosome. When we introduced that alien DNA into the genome, it has become a part of the chromosome, which has the ability to replicate. Obviously, chromosome has the ability to replicate, and in this process, the whole alien DNA also replicates. In a chromosome, there is a specific DNA sequence called as origin of replication, which is responsible for initiating replication. Origin of replication is that place where replication starts. So when we introduce it near the place of origin of replication, then it is obviously most likely that we get the desired DNA in the progeny. Therefore, for the multiplication of any alien piece of DNA in an organism, it needs to be a part of the chromosome which has a specific sequence known as origin of replication. Thus, an alien DNA is linked with the origin of replication so that this alien piece of DNA can replicate and multiply itself in the host organism. This is also known as cloning or making multiple identical copies of any template DNA. So, if you want your desired DNA to be expressed in the progeny, then what you should do, put that DNA, insert that DNA in the chromosome area where the origin of replication site is located. There, the alien piece of DNA can replicate and multiply itself in the host organism. This process is known as cloning because whatever you desired, that product is coming. So, this is known as cloning or making multiple identical copies of any template DNA. Let us now focus on the first instance of the construction of an artificial recombinant DNA molecule. The construction of the first recombinant DNA emerged from the possibility of linking a gene encoding antibiotic resistance with a native plasmid. Plasmid is autonomically replicating circular extrachromosomal DNA of Salmonella typhimurium. Stanley Cohen and Herbert Boyer accomplished this in 1972 by isolating the antibiotic resistant gene by cutting out a piece of DNA from a plasmid which was responsible for conferring antibiotic resistance. So, plasmid is autonomically replicating, means they do not depend on the DNA, they are the cells which replicate on their own. They have their own extra chromosomal DNA. They are present in a bacteria and they can replicate on their own. So, when Herbert Boyer and Stanley Cohen accomplished this in 1972, they isolated an antibiotic resistance gene by cutting out a piece of DNA from a plasmid. Plasmid contained DNA and that piece of DNA was cut, which was responsible for resisting that antibiotic action. This was cut and it was responsible for conferring antibiotic resistance. The cutting of DNA at specific locations became possible with the discovery of so-called molecular scissors restriction enzymes. The cut piece of DNA was then linked with the plasmid DNA. Then that cut piece was cut possible by molecular scissors which are known as restriction enzymes. Then that piece was linked with the plasmid DNA. The plasmid DNA act as vectors to transfer the piece of DNA attached to it. You probably know that mosquito acts as an insect vector to transfer the malarial parasite into the human body. In the same way, a plasmid can be used as a vector to deliver an alien piece of DNA into the host organism. If this is the desired DNA and we are cutting this plasmid from two ends with the help of molecular scissors, which are known as restriction endonucleases, then here we attach the desired DNA and this plasmid will now act as a vector. If this is a bacterial cell, this plasmid will be injected into the bacterial cell. This is acting as a vector, and when this plasmid is introduced inside, this DNA will start replicating, and that's why this bacteria will become transformed. So this was the explanation. Now, the linking of an antibiotic resistance gene with the plasmid vector becomes possible with the enzyme DNA ligase, which acts on the cut DNA molecule and join their ends. Here, when we were sticking the desired DNA back into the vector, here we use the enzyme DNA ligase. Ligase means to stick. So it basically sticks this DNA into the plasmid. This makes a new combination of circulus autonomous replicating DNA created in vitro and is known as recombinant DNA. When this DNA is transferred into Escherichia coli, a bacterium closely related to Salmonella, it could replicate using the host new hosts DNA polymerase enzyme and make multiple copies. The ability to multiply copies of antibiotic resistance gene in E. coli was called cloning of antibiotic resistance gene in E. coli. Here, after this foreign DNA incorporated into the plasmid, we called this recombinant DNA. So we transferred this recombinant DNA into E. coli, and this E. coli produced multiple copies of this desirable genes which had the DNA of our desired, and this process we name it as cloning. You can hence infer that there are three basic steps in genetically modified an organism. First one, identifying of DNA with desirable genes. Second, introduction of identified DNA into the host, maintenance of introduced DNA in the host, and transfer the DNA to its progeny. First, we have identified our desirable gene. What is the second step? We have to ensure that this desirable gene went into the host through a plasmid, a recombinant DNA. With the help of a recombinant DNA, our desired gene got incorporated into the host. And the third step is this is incorporated in the progeny. We should ensure that there are multiple copies being created in its progeny, which is known as cloning. So these were the three basic steps in genetically modifying an organisms. Now let's look at the tools of recombinant DNA technology. What are the equipments we need to recombinate the DNA technology? Now we know from the foregoing discussion that genetic engineering or recombinant DNA technology can be accomplished only if we have the key tools. First is restriction enzymes. Second, polymerase enzymes. Third, ligases. Fourth, vectors. And fifth, the host organism. Let us try to understand some of these in detail. Restriction enzymes. In the year 1963, the two enzymes responsible for restricting the growth of the bacteriophage in Escherichia coli were isolated. One of these added methyl groups to DNA while the other cut the DNA. The latter was known as restriction endonucleases. So in this diagram, when we were transforming a plasmid, we have cut it through a restriction enzyme, restriction endonucleases. The work of this restriction endonucleases is just to cut the enzymes, and we were isolating the desired gene from another organisms. We also used restriction endonucleases in this case also. The first restriction endonuclease, HIND2, whose functioning depended on a specific DNA nucleotide sequence, was isolated and characterized 5 years later. It was found that HIND2 always cut DNA molecules at a particular point of recognition, a specific sequence of 6 base pairs. This specific sequence is known as recognition sequence for HIND2. Besides HIND2, today we know more than 900 restriction enzymes that have been isolated from over 230 strains of bacteria, each of which recognize different recognition sequence. If we consider this to be a rough diagram of a DNA, it identifies a specific base pair, sequence of base pairs, and there are specific restriction enzymes. They recognize specific sequences of base pairs, and these sequences, they are known as recognition sequence. This could be a recognition sequence for HIND2. This could be a recognition sequence for another restriction endonuclease. This for another restriction endonuclease, etc. So all of them have specific recognition sequences, and they cut the DNA at only that particular point and nowhere else. So at present, we have 900 restriction enzymes which have been isolated from over 230 strains of bacteria, and HIND2 was the first restriction endonuclease to be discovered. The convention for naming these enzymes is the first letter of the name comes from the genus and the second two letters come from the species of the prokaryotic cell from which they are isolated. For example, EcoRI. This is the name of a restriction enzyme. So the first letter E in here, it comes from genus. The second two letters, they are the species. R, the name of a strain, and this Roman number is order in which they were isolated. So EcoRI or EcoR1 is actually, it comes from Escherichia coli RY13. So whatever the name of the restriction enzyme is there, the first alphabet represents the genus, the second two represent the species of the bacteria from which they were extracted, the third one, or you can say the fourth one actually, represents the strain of the bacteria, and the last Roman number indicates the order in which they were isolated. Restriction enzymes belong to a larger class of enzymes called nucleases. They are of two kinds, exonucleases and endonucleases. Exonucleases remove nucleotides from the ends of the DNA, whereas endonucleases make specific cuts at specific positions within the DNA. So nucleases is the larger class. This class is divided into endonucleases and exonucleases. Exonucleases remove nucleotide from the end of DNA, while endonucleases cut the DNA at specific position. And how do they do that? Each restriction endonucleases functions by first one inspecting the length of a DNA sequence. Once it finds its second one, finding the specific recognition sequence. Third one, it will bind to the DNA, and lastly, it will cut each of the two strands of the double helix at specific points in its sugar phosphate backbone. Each restriction endonuclease recognizes a specific palindromic nucleotide sequence. So, endonucleases cut the DNA at specific position. And what does it do? First of all, it inspects the length of the DNA. Second, it recognizes the recognition sequence. Third one, it binds to the DNA. And lastly, it cuts the two strands by breaking the sugar phosphate backbone. Do you know what palindromes are? These are the groups of letters that form the same word when read both forward and backward. Example, Malayalam. As against a word palindrome where the same word is read in both direction, the palindrome in DNA is a sequence of base pairs that read same on the two strands where orientation reading is kept the same. For example, the following sequence reads the same on the two strands 5 prime to 3 prime or 3 prime 3 prime to 5 prime. GAA-TTC, GAA-TTC. So these restriction enzymes, they cut the DNA little away from the center of the palindromic sites, but between the two base pairs of the opposite strands. This leaves single stranded portion of the DNA that are overhanging stretches called as sticky ends on the both strand. They are named so because they have hydrogen bonds with their complementary cut counterparts. The stickiness of the ends facilitate the action of the enzyme DNA ligase. In this diagram, we can say vector DNA is being cut by EcoRI restriction enzyme, and the foreign DNA is also cut. It is also cut by the same restriction enzyme, that is EcoRI. It cuts between bases of G and A. GAA-TTC is the recognition sequence in the base pairs. It cuts between that. When it is cut, there are sticky ends produced. Sticky ends means they have hydrogen bonds, and these sticky ends are then joined together by the help of another enzyme called DNA ligase. This is the steps in formation of recombinant DNA by the action of restriction enzyme EcoRI. Restriction endonucleases are used in genetic engineering to form recombinant molecules of DNA, which are composed of DNA from different sources or different genomes. When cut by the same restriction enzyme, the resultant DNA fragments have the same kinds of sticky ends, and they can be joined together end to end using DNA ligases. Here also we have this foreign DNA and the vector plasmid DNA. They are cut by the same restriction enzyme, so they produce same kinds of sticky ends. This is cut and this is cut, and they are joined together These are the sticky ends. You can see this end, and here this end, this ends. They have sticky ends, they have hydrogen bonds. They are joined together by DNA ligases. This is the vector DNA. It is transformed into E. coli gets transformed when vector DNA is introduced into E. coli and these cells divide. We have the desired progeny. This is diagrammatic representation of recombinant DNA technology. You may have realized that normally, unless one cuts the vector and the source DNA with the same restriction enzyme, the recombinant vector molecule cannot be created. Which means if we use different restriction enzymes for cutting the foreign DNA and a different enzyme for cutting the vector DNA, then vector DNA cannot be made. It cannot be created because we need to have same sticky ends for them to join. So same restriction enzyme will cut between same base pairs, so therefore they will have same sticky ends. Therefore it is very important for creating a recombinant vector, you need to cut the vector and the foreign DNA with the same restriction enzymes. Now, separation and isolation of DNA fragments. How are DNA fragments from the desirable, if you want some desirable gene to be transformed into a host, we need a vector in between. So from the first step, how do we take the DNA fragments out and how do we cut and isolate these? The cutting of DNA by restriction endonucleases results in the fragments of DNA. These fragments can be separated by a technique known as gel electrophoresis. Since DNA fragments are negatively charged molecules that can be separated by forcing them towards the anode using an electric field through a medium or matrix. Nowadays, the most commonly used matrix is agarose, which is natural polymer extracted from seaweeds. The DNA fragments separate according to their size through sieving action provided by the agarose gel, hence the smallest fragment size, the farther it moves. If this is a typical agarose gel electrophoresis showing migration of undigested, and so you can see the smallest one here, the largest one cannot travel most of the distance. The smallest one will be located near the anode and the largest one will be still left behind. The separated DNA fragments can be visualized only after staining the DNA with a compound known as ethidium bromide followed by exposure of UV radiation. First, you cannot look at the naked DNA. You need to stain the DNA with ethidium bromide. Then you need to uh, expose it to UV radiation, UV lights. You cannot see pure DNA fragments in the visible light, and without staining, you cannot see. So, when you expose it to UV light after staining it with ethidium bromide, you can see bright orange color bands of DNA in ethidium bromide stained gel exposed to UV light. The separated bands of DNA are cut out and the agarose gel and extracted from the gel piece. This step is known as elution. Means taking out the DNA from the agarose gel is known as elution. The DNA fragments purified in this way are used in constructing recombinant DNA by joining them with cloning vectors. Then when this DNA is removed through a process known as elution, this is transferred into the cloning vector and this helps in the creating of recombinant DNA. Now, what are the properties of cloning vector? How do we select a cloning vector? You know that plasmids and bacteriophages have the ability to replicate within the bacterial cell independent of the control of chromosomal DNA. Plasmids, they do not depend on the chromosomal DNA, they can replicate on their own. Bacteriophages are actually the viruses which infect the bacteria. So they can also reproduce on their own inside a bacterial cell. Bacteriophages, because of their high number per cell, have very high copy number of their genome within the bacterial cell. Some plasmid may have only 1 or 2 copy per cell, whereas others may have 15 to 100 copies per cell. Their number can go even higher. If we are able to link an alien piece of DNA with bacteriophage or plasmid DNA, we can multiply its number equal to the copy number of the plasmid or bacteriophage. So vectors used at present are engineered in such a way that they help easy linkage of foreign DNA and select of recombinants from non-recombinants. So what is copy number? Copy number is the number of copies they are producing. If the plasmid or the bacteriophage has a very high copy number, we select that as a vector, because if we have many copies, we have more capacity of them getting into bacterial cell, host cell, and them getting replicated into the progeny. These are the features that are required to facilitate cloning into a vector. First, origin of replication, or ORI. This is a sequence from where replication starts. We have already seen that origin of replication, name only is suggesting that from here the replication is starting, and any piece of DNA when linked to its sequence can be made to replicate within the host cell. If you have placed the DNA exactly on the ORI, then you have high maximum capacity that this DNA is going to replicate in the host cell. This sequence is also responsible for controlling the copy number of the linked DNA. So if one wants to recover many copies of the target DNA, it should be cloned in a vector where origin supports high copy number. Exactly, if this vector, it has high copy number, means it can produce many copies, then this vector should be selected. Its ORI also makes many copies. Therefore, the foreign DNA we should be placed near the ORI, and this vector should have high copy number. Second feature is selectable marker. In addition to ORI, vector requires a selectable marker which helps in identifying and eliminating non-transformants and selectively permitting the growth of the transformants. Transformation is a procedure through which a piece of DNA is introduced into the host bacterium. Normally, the genes encoding resistance to antibiotics such as ampicillin, chloramophenicol, tetracycline, or kanamycin, etc., are considered useful selectable markers for E. coli. The normal E. coli cells do not carry resistance against any of these antibiotics. Let's talk about cloning sites. In order to link the alien DNA, the vector needs to have very few, preferably single recognition site for the commonly used restriction enzymes. Presence of more than one recognition sites within the vector will generate several fragments, which will complicate the gene cloning. We have previously seen that this is the recognition sequence for one particular restriction enzymes. If this is the textbook is saying that a particular vector has only one recognition sequence. If we have more than one, if this is also the recognition sequence for that particular restriction enzymes, this will also be cut, and this fragment will also be cut. This will result in many fragments. So the characteristic of an ideal vector is it has only one recognition sequence, not many, so that this process becomes more complicated if many fragments will be generated. The ligation of an alien DNA is carried out by restriction site present in one of the two antibiotic resistance genes. For example, you can ligate a foreign DNA at the place of BAMH1 site of tetracycline resistance gene in the vector PBR322. Here, Here in this tetracycline resistance area, if we insert the foreign gene in the BAM H1, this will have the recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but can still be selected out of non-recombinant ones by placing the transformants on a tetracycline containing medium. This DNA is tied here, and if it gets transformed, then it will lose its capacity to resist tetracycline. To understand this, this is the rough diagram of PBR 322 plasmid. In here we are saying that we have transformed this in the BAM H1 region, which is tetracycline resistance. Now if we have two culture plates, this plasmid is transformed and it loses its ability for tetracycline resistance because we have inserted this foreign DNA in this area where tetracycline resistance was there. If we grow this transformed plasmid in ampicillin region, it will grow perfectly. And if we transfer it from ampicillin to tetracycline, it will not grow. This is the meaning of that it is a transformed vector. If we want to know whether it is transformed or not, first we will grow it on the ampicillin medium. If it is growing, then it's transformed back again into tetracycline medium. If it does not grow in tetracycline medium, means there is a DNA inserted in the tetracycline resistance which is resisting its growth in the tetracycline medium. So I hope you understood this statement. This is what is mentioned here. The recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but can still be selected out of non-recombinants one by placing the transformants on tetracycline containing medium. The transformants growing on ampicillin containing medium are then transferred on a medium containing tetracycline. The recombinants will grow in ampicillin containing medium but not on containing tetracycline. But non-recombinants will grow on medium both, because they do not have a gene inserted in their tetracycline resistance area. Both the antibiotics. In this case, one antibiotic resistance gene helps in selecting the transformant, where the other antibiotic resistance gene gets inactivated due to insertion of alien gene and helps in selection of recombinants. In this case, when we have planted this foreign DNA in tetracycline resistance antibiotic, ampicillin was helping us, and the tetracycline got inactivated because there were the foreign DNA implanted in it. Selection of recombinants due to insertional inactivation. Selection of recombinants due to inactivation of antibiotics is a cumbersome procedure because it requires simultaneous plating on the two plates having different antibiotics. Therefore, alternative selectable markers have been developed with differentiate recombinants from non-recombinants on the basis of their ability to produce color in the presence of chromogenic substrate. In this, a recombinant DNA is inserted within the coding sequence of an enzyme beta-galactosidase. This results into inactivation of the gene from synthesis of the enzyme, which is referred to as insertional inactivation. The presence of the chromogenic substance gives blue-colored colonies. If the plasmid in the bacteria does not have an insert, presence of insert results into insertional inactivation of the alpha galactosidase, and the colonies do not produce any color. These are identified as recombinant colonies. In this method, insertional inactivation, we differentiated transformants from non-transformants, because the transformants did not produce any color, and the non-transformants produced colors. Next, vectors for cloning genes in plants and animals. What are the common kinds of vectors we choose? You may be surprised to know that we have learned the lesson of transferring genes into plants and animals from bacteria and viruses, which have known this for ages. How to deliver genes to transform eukaryotic cells and force them to do what bacteria or viruses want. For example, Agarobacterium tumefaciens, a pathogen of several dicot plant, is able to deliver a piece of DNA known as tDNA to transform normal cells into a tumor and direct these tumor cells to produce the chemicals required by the pathogen. Similarly, retrovirus in animals have the ability to transform normal cells into cancerous cells. A better understanding of the art of delivering genes by pathogens in their eukaryotic hosts has generated knowledge to transform these tools of pathogens into useful vectors for transforming genes of interest to humans. Initially, what happened is Agarobacterium tumefaciens was a pathogen, and it it was causing a tumor in the plants. So instead of causing the tumor, we used it in benefit for our gene transfer into the plant. And in the case of animals, we have retrovirus, which was causing normal cells to become into cancerous cell. We removed that genes which transferred cancer, and we transformed a desirable gene into that. So if this retrovirus infects the animals, then instead of cancer, the desirable genes which we have introduced into the retrovirus, that will be transformed into the animals. This way, we have figured it out. The tumor-induced TI plasmid of Agarobacterium tumefaciens has now been modified into a cloning vector, which is no more pathogenic to the plants, but it's still able to use the mechanism to deliver genes of our interest into a variety of plants. See, we have modified the Agarobacterium tumefaciens. It is no more pathogenic. It no more causes tumor, but it helps in delivering our gene of interest into the plants. Similarly, retrovirus have also been disarmed, means it has become uh, it does not cause cancer anymore, and now it is used to deliver the desirable genes into animal cells. So once a gene or a DNA fragment has been ligated, means sticked into a suitable vector, it is transformed into the bacterial plant or animal cell where it multiplies. Next, we have competent host for transformation with recombinant DNA. How do we choose a host? Since DNA is a hydrophilic molecule, hydrophilic means water loving, it cannot pass through cell membranes. Why? In order to force bacteria to take up the plasmid, the bacterial cell must first be made competent to take up DNA. This is done by treating them with a specific concentration of a divalent cation such as calcium, which increases the efficiency with which the DNA enters the bacterium through pores in its cell walls. Recombinant DNA can then be forced into such cell by incubating the cells with recombinant DNA on ice, followed by placing them briefly at 42 degrees heat shock and then putting them back to ice. This enables the bacteria to take up the recombinant DNA. This is the bacterial cell and this is the vector, and we want this vector to get inside the bacterial cell, but the cell wall is the problem. So we treat this bacterial cell with a divalent cation such as calcium. This will increase the efficiency. I mean, uh, this will create pores between the cell wall, so this plasmid can enter through this pores. There is another method known as heat shock. First, we incubate the cells in ice, first in extreme cold, then in heat 42 degrees Celsius heat, then again back in ice. This method is known as heat shock method, and this method will force bacteria to take up the vector. So there are two methods to transport this vector into the bacteria. This is not the only way. It is introducing us to another way also, to introduce the alien DNA into the host cell. In a method known as microinjection, recombinant DNA is directly injected into the nucleus of an animal cell. In another method suitable for plants, cells are bombarded with high velocity microparticles of gold or tungsten coated with DNA in a method known as ballistics or gene gun. And the last method uses disarmed pathogen vectors, which, when allowed to infect the cell, transfer the recombinant DNA into the cell. So microinjection is a method in which directly we are injecting the recombinant DNA vector into the animal cell, and microinjection is suitable for animal cell. While talking about plants, we use a method known as biolistics or gene gun, in which we have high velocity of cells which are coated with DNA, particles of gold or tungsten which are coated with DNA, and they are bombarded, they are fired on the plants. Now we have learned about the enzymes, restriction endonucleases, DNA ligases, etc. We have also seen how restriction endonucleases will cut the DNA at specific recognition sites and recognizing the palindromic sequence. We have also seen gel electrophoresis, in which we can separate and isolate the DNA fragments. We have seen the characteristics of cloning vectors and what are the suitable things we look up when we are choosing the cloning vectors. We have also seen how we choose a competent host, and we have also seen the methods of transforming the vector into the host cell. Now we will learn about the process of recombinant DNA technology, how this process is brought about. Recombinant DNA technology involves several steps in specific sequence, such as isolation of DNA fragmentation of DNA by restriction endonucleases, isolation of desired DNA fragment, ligation of the DNA fragment into the vector, transferring the recombinant DNA into the host, culturing the host cell in a medium at large scale, and extraction of the desired products. Let us now examine each of these steps in some detail. First of all, we have to choose desired gene. So for that, we have to take out the DNA of that desired gene. Recall that nucleic acid is the genetic material of all organisms without exception. In majority of the organisms, that is deoxyribonucleic acid or DNA. In order to cut the DNA with restriction enzyme, it needs to be in the pure form, free from other macromolecules. Since the DNA is enclosed within the membranes, we have to break the cell open to release DNA along with other macromolecules such as RNA, proteins, polysaccharides, and also lipids. This can be achieved by treating the bacterial cell or plant cell or animal tissue with enzymes such as lysozyme for bacteria. Since see, the cell wall of bacteria is made up of various materials, and we lysozymes dissolve the cell wall of the bacterial cell. So in order to dissolve the cell wall, we use lysozyme for bacteria. The plant cell is made up of cellulose. To dissolve cellulose, we need the enzyme cellulase. Cellulase acts upon the cell wall and breaks it. For fungus, if we want to isolate the DNA of the fungus, the cell wall is made up of chitin. So to destroy chitin, we need the enzyme chitinase. Now, you know that genes are located on long molecules of DNA intertwined with proteins such as histones. These histones and the DNA is packed together, and the genes are located in the DNA. The RNA is also present in the DNA. So this RNA can be removed by treatment with ribonuclease. When we treat RNA, that is the ribose nucleic acid, by ribonuclease, RNA gets destroyed. Proteins are also present. Now these proteins can be removed by treatment with protease. Other molecules can be removed by appropriate treatment, and lastly we get the purified DNA, and this is precipitated out after the addition of chilled ethanol. This can be seen as a collection of fine threads in the suspension. This is a diagram showing DNA that separate out removed by spooling. Spooling is a method in which we are removing the DNA out after adding chilled ethanols, and this can be seen as fine threads. The next step we have is cutting the DNA at specific location. We have removed the cell wall, we have removed the RNAs, we have removed the proteins, we have all obtained the pure form of DNA by spooling method. Now we have to cut this pure DNA. Restriction enzymes, obviously the restriction enzyme comes here into the picture. Restriction enzymes digestion are performed by incubating purified DNA molecules with the restriction enzyme at optimal conditions for the specific enzyme. Agarose gel electrophoresis is employed to check the progression of the restriction enzyme digestion. We have seen the electrophoresis process. DNA is negatively charged, hence it moves towards the positive electrode anode. The smaller the DNA particle, the more near the anode it will be present. So in this gel electrophoresis, we will then stain it with etherium bromide and project UV radiations. Then we can see the DNA. Then the process is repeated with the vector DNA also. We cut the vector DNA also with the same restriction enzymes. Now, the joining of DNA involves several processes. After having cut the source DNA as well as the vector DNA with specific restriction enzymes, the cut out gene of interest from the source DNA and the cut vector with space are mixed up and ligase is added. This results in the preparation of recombinant DNA. In this diagram, we have already seen that we are cutting the foreign DNA as well as the vector DNA, that is plasmid, with the same restriction endonuclease. Here we can see it is cut, then it is joined by using ligase enzyme, and this, after it is joined, it is known as recombinant DNA molecule. Now, amplification of gene of interest using PCR. PCR stands for polymerase chain reaction. In this reaction, multiple copies of the genes of DNA of gene of interest is synthesized in vitro, in the lab or in the test tube, using two sets of primers. small chemically synthesized oligonucleotides that are complementary to the regions of DNA, and the enzyme DNA polymerase. The enzyme extends the primers using the nucleotides provided in the reaction and the genomic DNA as templates. If the process of replication of DNA is repeated many times, the segments of DNA can be amplified to approximately billion times. That is, 1 billion copies are made. Such repeated amplification is achieved by the use of thermostable DNA polymerase isolated from a bacterium, Thermus aquaticus, which remains active during the high temperature induced denaturation of double stranded DNA. The amplified fragment, if desired, can now be used to ligate with a vector for further cloning. Let's look at the polymerase chain reaction PCR. There are 3 steps each cycle. If you want one copy of the DNA, these 3 processes should be over and over. The first process is denaturation. The double stranded DNAs, you can see this undergoes heat. What happens is denaturation means separation of strands. There are 2 strands. DNA is double stranded. So first process, we separate the strands. It is separated. Then second step, it is kept in a very hot medium, 45 degrees Celsius. And in this step, annealing, we attach primers. Primers means oligonucleotides. We have seen here the definition of primers. They are small chemically synthesized oligonucleotides that are complementary to the regions of the DNA. That means they can directly bind to the regions of the DNA. They are nothing but chemically synthesized oligonucleotides. Here you can see primers are joined at specific ends. Now, then we introduce tuck polymerase. This is an enzyme. This can work even at high temperatures. Even at 72 degrees, it can work. Third process is extension. This DNA strands are extended, and after 30 cycles you will get amplified DNA. This is process is known as amplification. 1 billion times, if it is repeated, you get 1 billion copies. So these are the steps of PCR polymerase chain reaction. First one is denaturation, where we have separated the strands. Second is annealing, where we are adding or attaching the primers. Primers are nothing but chemically synthesized oligonucleotides. Then we are introducing an enzyme which is known as tuck polymerase. This can even work at very high temperature, and this tuck polymerase is obtained from a bacteria. We have read it here. Thermus aquaticus. This is isolated from a bacterium, Thermus aquaticus. This is thermostable. Thermo means temperature. Stable means it is stable at high temperatures. Then the next step we have is extension, in which the DNA strands are extended. Then the last one is amplification. That is nothing but repetition of that cycle. If it is repeated 1 billion times, you get 1 billion copies. This is the mechanism of amplification of gene of interest using PCR. Next step is insertion of recombinant DNA into the host cell. There are several methods of introducing the ligated DNA into recipient cells. Recipient cells, after making them competent to receive, take up DNA present in their surroundings. So if a recombinant DNA bears gene of resistance of an antibiotic, that is ampicillin, is transformed into E. coli, the host cell becomes transformed into ampicillin resistant cell. If we spread the transformed cells on a agar plate containing the ampicillin, only transformants will grow. Untransformed recipient cells will die. Since due to ampicillin resistance gene, one is able to select a transformant cells in the presence of ampicillin. Here the ampicillin resistance gene in the cells is called a selectable marker. It helps us to select the recombinants and the non-recombinants. Next, obtaining the foreign gene product. When you insert a piece of alien DNA into a cloning vector and transfer it into a bacterial cell, that is our host, or a plant or an animal cell, the DNA gets multiplied. In almost all recombinant technologies, the ultimate aim is to produce a desirable protein. Hence, there is a need for recombinant DNA to be expressed. The foreign gene gets expressed under appropriate condition. The expression of foreign genes in host cell involves understanding many technical details. After having cloned the gene of interest and having optimized the condition to induce the expression of the target protein, one has to consider producing it on a large scale. Can you think of any reason why there is a need for large scale production? Because since in the first introduction we have learned that biotechnology means human useful. If anything, if any product is useful for the humans, we are producing it through biotechnology. So there are billions and billions of humans in this world. So if these humans need to get benefit, we need to produce it on a large scale. Therefore we produce it on a large scale. It is necessary. If any protein encoding gene is expressed in a heterologous host, it is called recombinant protein. The cells harboring cloned genes of interest may be grown in a small scale in a laboratory. The culture may be used for extracting the desirable protein and then purifying it by using different separation techniques. The cells can be multiplied in a continuous culture medium wherein the used medium is drained out from one side and fresh medium is added from the other to maintain cells in their physiologically more active or exponential phase. This type of culturing medium produces larger biomass leading to higher yields of desired protein. Small volume cultures cannot yield appreciable quantities of products. To produce in large quantities, the development of bioreactors where large volumes, about 100 to 1000 liters of culture, can be processed. Thus, bioreactors are considered to be large vessels where the raw materials are biologically converted into specific products, individual enzymes, etc. These are two kinds of bioreactors. This is the most common one, and this is known as simple stirred tank bioreactor. This is pranked stirred tank bioreactor. Now, what are the conditions? There, these bioreactors, they help to maintain the optimal conditions for achieving the desired products. They make optimum growth conditions. They check the temperature. They check the pH, substrate, salts, vitamins, and oxygens of all their surroundings in order to get the desired products. This stir-tacked bioreactor is special because it is usually cylindrical and it has a curved base to facilitate the mixing of the reactor contents. And the stirrer, this it helps in stirring and continue availability of oxygen throughout the process. Now we have the last step, which is known as downstream processing. After completing of the biosynthetic phase, we need to check this product whether it is ready for marketing. Downstream processes means we are separating and purifying the products which we have obtained. We have to check with the suitable preservatives whether it could be preserved for a longer period of time. And if we have produced drugs, we need to check clinical trials. We need to check on what patients it is advisable or not. We have to also check the quality of the products. So this is the end of chapter 11, biotechnology principles and processes, in one shot. If you have any doubts, you can comment down below, like the video, share it with your friends and subscribe to the channel. Thank you.